Welcome to prokaryotic transcription land. Transcription is how you get from DNA to RNA. The enzyme RNA polymerase copies the DNA strands by making phosphodiesterase bonds in 5' to 3' direction to join nucleotides together. Phosphodiesterase bonds are a bond between two sugar groups and a phosphate group that form the backbone of DNA and RNA. RNA polymerase does not require a primer to begin synthesis. Subunits of RNA polymerase are core enzyme and holoenzyme. In this lane, we focus on the holoenzyme because it has all factors, including the sigma, while the core does not. The sigma factor can change features of RNA polymerase, making it vital to the overall outcome. DNA and RNA must be antiparallel. Non-template DNA can be called the coding strand or sense strand. This goes in the 5' to 3' direction. The RNA strand that follows the coding strand is called the template strand. The template strand goes in the 3' to 5' direction. A consensus sequence is a segment where popular nucleotides are expressed based on another particular segment. In prokaryotic transcription, we have two consensus sequences of importance, the -10 and the -35. The -10 is often called the Pribnow or the Tata box, while the -35 is called the -35 control. The promoter region is home to both. Since so many prokaryotes have them both, it suggests that they serve a purpose. This purpose is the recruitment of RNA polymerase. RNA polymerase binds DNA at consensus sequences in order to prepare the RNA polymerase for transcription initiation. Protein-DNA interactions occur at both the sites, which is why they are able to serve this purpose. In the very end of the promoter region, there lies the +1 nucleotide where transcription begins. The promoter region is where RNA polymerase recognizes the initiation of transcription. +1 serves as a reference point, determining if a nucleotide will be located upstream or downstream. Once the DNA strands separate to form the bubble at +1, transcription can begin, with RNA polymerase binding at the promoter thanks to sigma. In the case of prokaryotic transcription land, there are inverted repeats. These are repeated sequences of RNA that eventually lead to the termination of transcription through hairpins. Termination hairpins are stem loop structures in the RNA that cause the end of transcription or polymerase pause. These hairpins are followed by a poly-A tail on the template strand on DNA, A for adenine. In prokaryotes, either row or hairpins are usually required for termination. Since the AU bonds are weak, the DNA-RNA bond will quickly unwind due to helicase activity. What would happen if the -10 box is deleted in a gene that codes for an enzyme to metabolize sugar? In this case, the RNA polymerase, or segment of interest here, is a gene that allows for enzymatic production, in turn breaking down sugars. The nucleotides would no longer be expressed for that gene, and the coding strand would not be transcribed to metabolize sugar because of its deletion. Thanks for watching.